Dominic Furniss, going to present the genetics of Dupuytren disease, which is the starting point of everything that we're doing here. Thanks, Dom.
Thank you, Charlie. So I'm Dominic Furniss. I'm a professor of plastic surgery from Oxford University. I have a sort of conflict of interest. I'm funded, at least in part, by the UKRI, which is the Medical Research Council in the UK, to run something called the Human Functional Genomics Initiative, the MSK cluster, and I'll tell you a little bit about that later on. Fantastic. Okay. So I'm going to start off with a controversial statement, and then I'm going to go into a little bit about why I think that that's the case, and then we'll talk about genetics and where we're going next. So, my controversial statement, which I don't think is quite so controversial because Charlie's kind of already made it for me, is that surgery is a failure in chronic disease. If we look back 30 or 40 years, we used to do operations like vagotomies and even gastrectomies for people with peptic ulcer disease, until Barry Marshall, Robin Warren from Australia recognized that this was actually an infection of H. pylori, and if you got rid of that infection and you gave antacid treatment, you didn't need to cut the stomach out. Similarly, as a hand surgeon, when I was at medical school, we used to do a combined clinic of hand surgery and rheumatology every week, and we would put 4 or 5 people on the operating list for their rheumatoid hand surgery. And we would do things like these MCP joint replacements. And now, as a consultant hand surgeon, I barely do any operations at all on patients with rheumatoid arthritis because of these new treatments, these biological treatments. And surgery, therefore, I think in chronic disease is a failure to understand the biology of the disease. In the case of rheumatoid arthritis, the inflammatory pathways around, particularly centered on TNF. And Dupuytren disease is a typical chronic disease, as we've already heard from Charlie. And therefore, I think in Dupuytren disease, surgery is a failure. So how do we understand the biology of a complex disease, a chronic disease? Well, we know that it's caused by an interaction of genetic factors and non-genetic factors, and you can take these different approaches, studying the genetics, epidemiology, the cell biology, and I'm going to focus on the genetics. So we have this interlinking between these genetic predisposition and the non-genetic stressors which play on that predisposition to give us the disease that we see in our patients. And we've looked, and there are a whole heap of other papers, but we've looked in my group at some of the non-genetic factors that are associated with Dupuytren disease, and I think there's been a bit of an emphasis so far this morning on mechanical stress, and certainly we see that work and occupation is a definite predisposition to getting Dupuytren disease as well. But what about the genetics? Well, the reason you look different to the person sitting next to you is because you have a whole heap over around 10 million different genetic variants in your genome compared to the person sitting next to you. And some of these are rare, and they have a big effect on how you look and what diseases you might have. And some of them are really common, and they have a very small effect on how you look and the different diseases that you get. And some are in between, and they may be very uncommon, and they have a sort of intermediate effect on what the expression of a particular disease or a trait is. And we use different methods to discover what these variants are. So for common variants, these are the ones most likely implemented in common diseases like Dupuytren's. We use something called a genome-wide association study. And for the low-frequency variants, we use something called an exome-wide association study. And I think we're all in agreement. This was controversial 15 years ago in Miami, but I think we're all in agreement now that Dupuytren's is not a Mendelian disease at all. So what's a genome-wide association study? Well, remember I said we've all got those 10 million, roughly, variants? Well, the vast majority of those are things called single nucleotide polymorphisms, or SNPs, and we all inherit one copy of our DNA from our mother and one from our father. And there are lots of places through the genome where the particular genetic letter can vary. And so in this example, you may inherit an A from your mother and an A from your father, in which case your genotype is AA. You may inherit an A from one and a T from the other, in which case it's AT, or you could inherit two Ts. And in a genome-wide association study, what we do is we look at that genetic code across around about 1 million different places in the genome, and we compare the frequency of those two different letters in cases and controls. Now, because we're doing 1 million tests, we need to make sure that our p-value is corrected for that, and so we use a p-value of 5 times 10 to the minus 8, so that's 0.00005. I think I got the number of noughts right there. And so for that, in order to be powered to get that level of significance, we need thousands of cases and thousands of controls. But this gives us a very unbiased view of where in the genome there might be predisposition to a particular disease. So this is the workflow. We collect data, we genotype, we do quality control, we do something called imputation. Then we do those association tests, and we get this kind of plot that you can see in the middle on the right, which is called a Manhattan plot, and that goes by chromosome and tells us the areas of the genome where we should be looking for some genetic predisposition. You can then meta-analyze these studies, you can replicate them in other cohorts, and there's a whole heap of post-GWAS analyses that have been done. XWAS is very similar, apart from we only then focus on the protein-coding genes within the genome. And so that's only about 1% of our genome. And the reason that we do that is because if there is an effect on a particular protein, rather than it being affecting protein levels or something like that, it's likely to have a higher impact on the organism. And we do two different kinds of analyses here. We do variant level analyses and we do gene-based analyses, because you could have one variant in a gene from your mother and one from your father, and that could compound and cause an effect. So what's happened in Dupuytren's disease? It goes back to Miami. So thank you, Wolfgang, and thank you, Charlie. I think the reason that I'm standing here today is because of this conference in Miami, where I met Paul Verka, who you'll all hear speak soon, and his PhD student Guido Dolmans, who were running the first genome-wide association study in Dupuytren's disease, and also Hans Hennies, who was trying to do a very similar thing in Germany. And this was at the stage where I was just collecting my cohort in the UK as well. And in the spirit of cooperation and collaboration, we all came together in order for us to be bigger than the sum of our parts. And we produced the first genome-wide association study on Dupuytren's disease, which was published in 2011. And we found 9 areas in the genome which were associated with Dupuytren's disease. And you've heard talk about the Wnt signaling pathway already. That came up as a very strong predisposition here. And since then, there have been multiple further studies. So we published again in 2017, where we increased the number of loci that were associated. And there have been 2 more recent studies in the last couple of years, where we now are up to around about 60 or so loci in Dupuytren's disease. And this is the GWAS catalog, which is freely available online. These are the hits plotted out in a Manhattan plot. Why should you care about this? You could say genetics has been useless. You've taken 15 years, and there's still no new treatments for my patients with Dupuytren's disease. I still have to do surgery, which you've already told me is a failure. So why should I care? And if I look across musculoskeletal diseases in general, there are tons of loci, and the treatments that we have are still mainly painkillers, physiotherapy, surgery, maybe radiotherapy for Dupuytren disease, and possibly for some of the other conditions as well. Well, the reason that you should care is that genetic targets are much more likely to lead to successful drugs, and there have been several studies on this, and I've just picked out one of them, where if you look at the right-hand side of each of these graphs, where if we're looking for a specific trait, a specific disease, a genetic-led hit from a GWAS is more than twice as likely to lead to a successful drug getting FDA approval. And if you have something that's more deleterious, so perhaps from the XWAS, then it's up to 4 times more likely. So this is a really important starting point. But this transformation, I think, requires some other things. So first of all, I think it requires human tissue. So finding out how this genetic variation leads to changes in the affected tissues in our patients, I think is critical. And as people have mentioned already today, I think we need physiologically relevant stressors when we're doing human disease models. So what you can see here is from one of my collaborators in my department. This is a humanoid bioreactor of the shoulder joint, modeling frozen shoulder. So at that shoulder joint, there's a tiny little bioreactor where we can put shoulder capsule in, and we can vary the media that's going through, and we can keep the tissue alive for a couple of weeks. And we can subject it to relevant mechanical stressors. And we're currently building a finger model just like this for Dupuytren's. And the whole point really is to go down this experimental medicine pipeline of going from basic science research, translating it into humans, and then into patients, and eventually out into practice. But you'll notice this early phase is called the Valley of Death. And that's where I think we can really have a big impact, is defining from our genetics and from our other functional studies, what is the target? What is the key target here? And then we can look in our lab-based studies and define what the intervention is. So, what the outcome we're going to measure, what the local biomarker is. So how does a drug affect the expression of some of these genes, for example? And then I think the key first step is to test that drug in a small patient sample. And that's why we as surgeons have a very special role, because we can give a drug to our patients for 1 week or 2 weeks or a month before they have their fasciectomy surgery. And then we can look in those samples to say, did that drug have the effect that we were expecting? And so I'm very fortunate to have been funded to do this exact pipeline across 4 different musculoskeletal diseases, so osteoarthritis, Dupuytren's, frozen shoulder, and carpal tunnel syndrome, where we'll be following that pathway and trying to identify and validate new targets in the coming few years. And the idea, as I said, is to take it from that genetic discovery through the validation step, ex vivo validation in human tissues, and try to get a drug into patients in the next 5 years. So I hope over the next 15 years of coming to these meetings, we will make that leap from being a failure to being a success. Thank you.
That was wonderful. Thank you so much. Questions? I'll start with a question. So ultimately, at least a quarter of the European population has Dupuytren latency. If you look at senior men, about a 25% incidence. So the question is, why doesn't everyone get Dupuytren? Is the genetic risk, is it actually a loss of function or loss of protection, like I was talking about with hair follicles? And if that's so, does that change the approach to looking at the gene risk profile, whether you're looking for something which causes disease or simply something which simply prevents it? Or is that even a reasonable concept?
No, it is a reasonable concept. I would say the expression of Dupuytren's, as I said, is a combination of your genetic risk and your environmental exposure. So, and we're currently looking at the interaction between those two things, particularly with work exposure. Your idea about is there a genetic protection, I think, is a really good one. And it's something the drug companies are really interested in. So are there rare variants which mean that certain people never get a disease? Right. You know, is there something that switches off a particular gene in people, which means that they are protected from a particular disease? And the reason they're interested in that is it's relatively easy to switch off a protein compared to activating it. So they're really interested in those exact variants, and that's not something that we've been done. You probably noticed I kind of skipped over XWAS because it's not really been done yet for Dupuytren's. So I think that is a really important next step.
Morning. Why did you choose a study at a value of 5 times 10 to the minus 8 for the p-value for a test being significant? It's almost the same as what we use in high-energy physics.
Yeah, so it uses a relatively conservative statistical method called a Bonferroni correction, where you divide your nominal p-value, which is 0.05, by the number of tests that you're doing, which is effectively 1 million. So you then go from 5 times 10 to the minus 2 to 5 times 10 to the minus 8. You add six zeros.
I have another, oh, never mind.
Just with like the genetic loci that you found, I was just wondering, like, could that be used for like a genetic risk score for patients in terms of like prognosis that gets severe disease?
Yeah, that has been studied, particularly by Paul's group and some of his PhD students, and we know that it correlates with earlier onset, more aggressive disease, more likely to suffer from recurrence as well. It's not quite hit the level yet where we can use it clinically, I would say. And there's still, the clinical features still are more predictive of what your clinical course is going to be than your genetic risk as things stand.
We're going to move on.